So moving away from the historical context of developmental biology, let's have a look at some of the model systems, and let's talk about model systems. So developmental mechanisms have been studied in a few species, and these well-characterized species become then models for studying developmental biology. The choice of models, of organisms to study, um, have been both biological and historical. As we will see, some of these models have been chosen by perhaps chance has played a big role in the decision, but generally if they are easy to work with, they are more accessible models to study development. Um, they have been kept. In the beginning, the older models were perhaps because they had large eggs that were easy to study in the absence of high-powered microscopes, which is less of a limitation nowadays. Things that are important with models is that they have easy access to large numbers of eggs and embryos. If they are synchronous, it's an advantage, so that you have many eggs at the exact same stage. They're synchronous. That will be an advantage to you. You can do multiple observations in the same experiment. A good model should have good opportunities for experimental manipulation, and this can be in part that, for example, the embryo is very large, but at the same time you need to have large numbers of those, so not tiny eggs. But also genetic analysis, and this so being able to breed them in captivity is important. This is guided whether a species would be a successful model, right? And then if it has been a successful model, easy to breed, has a good sized embryo, produces a large number of eggs, then these are often chosen to continue working with, because there's a lot of knowledge being established that you don't need to re-characterize and redo experiments. You can build on the back of those other experiments. The classical models have largely been frogs or other amphibians, salamander and stuff, but also ascidians, tunicates, and sea urchins. We can mention that comparative studies between closely related models can reveal conservation and help identify the most basic mechanisms. If we look at very early animal ancestors, for example, we can compare several very early to see what was the new thing that was found between those very closely related animals, or we can look at more evolved animals such as us and very distant animals from the sea, for example, that have much smaller genomes, and we can see what steps has the genome taken during development, how much more complex it has been. And we'll hear more about this comparative work and the evolutionary aspect of developmental biology from Powell. I'll just add a few things to the choice of a good model for studying developmental processes nowadays. And it's very important that they are suited to genetic screening. So they should have a fast life cycle, a low cost to maintain, and it's a benefit if they have visible phenotypes. And that was more true perhaps in the old days with lower powered microscopes and less penetrating microscopes compared to what we have now. But these still hold true. And then you can think, okay, well, what's a short life cycle? Would you think an elephant has one? It lives for 90 years or something? And no, not really. So that's why we tend to work with smaller animals. The fruit fly has been a very popular favorite, but also the worm, C. elegans, Cenorhabditas elegans. These are both invertebrate, right? They don't have a backbone or as a complicated nervous system as vertebrates, such as the fish, is a step towards mammals, or of course we have rats and mice, which are more closely related to humans, and so in some ways serve as a much better model than, for example, the fly. But it depends on exactly what you're looking at. Also, of course, you have an elephant, the cost of food and maintenance is going to be much higher, right? So another question that has been very important is, are all stages of development possible to image? Can we monitor what's going on at all stages of development? And I would, to a certain extent, all of these models you can, but for example, with the mice, the embryos are still in the mother, so they need to be removed traditionally if you want to look at them. Whereas the fly, the worm, and the fish, they will lay their eggs, and we can collect their eggs after they've been laid and start looking at the earliest stages of development. That's one aspect of it, but you have to also then think a bit about if we're using what kind of microscopes we're using, and if we're just using normal transmitted light, the egg itself should be visible through the microscope. You should be able to look at what's going on at the cell divisions and those kind of things. So I just wanted to compare the perhaps 4 most popular model systems in use today. If we take the mouse, it has, I've given plus and minuses to these things. It has a 50-day life cycle. So experiments that take several generations to get to your mutant or study your phenotype will take a long time. If you want to make a knockout mouse, for example, it can take up to a year. Also, the genome is, of course, more related to the human genome, which is a benefit. But throughout these times, the genome has, from fly, for example, to mammals, there has been an extra duplication of the genome. So there are many more homologs of genes or paralogs in the animal, in the more evolved animal, than in the fly. So the fly might have just one gene for a protein in a certain pathway, whereas the mouse might have 2 for that. The fish, I just mentioned it now, that it has had an additional round of genome duplication, so that problem is made worse, that there are more paralogs for the genes, so it's harder to figure out what gene is the one to target, and what gene is important in the processes that you're interested in. I just mentioned that there. So there's more redundancy in these animals compared to these, more gene duplications and more redundancy between proteins. So the imaging with the mouse and the genetic approaches have been pretty low tech. They are improving now with the advent of CRISPR Cas9 system for making targeted gene deletions, but up to most recent times, this has been a very lengthy process to make mutant animals, for example. And the imaging possibilities, as I mentioned, with the microscope for development purposes, the embryos develop inside the mother, so it's not that easy to image them. They are expensive to keep. You need very highly trained staff. You need conditions that are very stable to house a large number of animals at the same time, but keep the animals in a very stress-free environment. So this is a challenge. So it costs a lot, it takes a lot of space. And then there are ethical considerations to all experiments that you do with vertebrate animals, including fish and mice. So every experiment that you want to do, you have to apply for ethical approval, which can take some time, and state the nature of your business, why you want to do this, why it's important to get permission to do your experiments. And you might be told no. So these are in a way I would consider these as negative aspects to working with these models. But there are pluses. You can freeze the embryos of these animals, and this isn't true for all of the model systems. And of course, they're a mammal, and work done in a mammal system will be accepted more readily, perhaps, than work done in an invertebrate system when it comes to certain diseases, for example. And if we just weigh up the pros and cons of working with fish, for example, zebrafish has a 90-day life cycle, so it's not the fastest in terms of screening. But imaging the embryos, remember that the eggs are laid outside and then fertilized after, and the imaging possibilities through the eggshell are fantastic. You can actually monitor all the divisions with a normal light microscope. The genetics for the fish, they are improving, but for example, CRISPR-Cas9 has also come to all of these organisms. So that improves the feasibility of doing mutant studies, generating mutants for particular genes. So the genetics are improving for fish, but traditionally they haven't been that good. And I mentioned that the genome has more duplications than mammals, so that's a complication when it comes to finding out which gene is doing what thing. It's not as intuitive as in some other organisms. They are relatively expensive to hold in captivity, and they take quite a lot of space, but this can be minimized to some extent by freezing down stocks, for example. There are ethical considerations, just like with the mouse. You have to apply for the rights to do your experiments and justify those. But you can also freeze the eggs, which is a big plus, and they're a vertebrate. So they're not a mammal, but they're a vertebrate, and they're therefore more closely related to humans and would be perhaps considered more relevant for studies on human diseases. Now, if we come to Drosophila melanogaster, which I work with, it has a very short life cycle, 10 days turnaround time from the egg being laid to having a fertile adult. Imaging is possible at all stages, and live imaging is possible at the majority of stages. And yeah, at this stage, you can say it's possible to image live at all stages, but more easier and difficult with depending on the stage and process you want to look at. They are very genetically amendable, as in manipulatable, very easy to manipulate the genetics of the fly. And they've been worked on for 100 years, and the genetics has improved over time. They have had the highest tech in terms of genetic tools Drosophila, the fruit fly, has had. And that still holds true because even though CRISPR-Cas9 has come to these other systems, it has also come to the fly. But to be honest, we have mutants for almost every single gene in the genome, and there are stocks of these available that other people maintain, and we just order them in. They are cheap to run. There are no real space requirements. You need a dedicated room to work with them, but that's true for all of these animals. Any genetically modified animal, you need a dedicated space in which to work with them. There are no ethical concerns when it comes to working with insects. They don't have a backbone, and you don't need to apply for approval to do any of your experiments. So these things speed up the rate at which you can produce results from these animals. But a large negative has to be mentioned. You cannot freeze any stage of the fly and defrost it and start your experiments again. So they need to be maintained. But they are cheap to run, so the food that they eat is very cheap, and they don't need that much care and attention. There are other negatives. Okay, I mentioned they didn't have a backbone, so they're not as related to humans. Some of the studies would not be as easily accepted as being relative to human diseases. But as I mentioned, I mean, most cancer pathways, for example, are conserved in flies, and not only are between flies and mammals, not only are they conserved, they were first found and studied in flies actually, and that was true primarily the study of development, looking for developmental defects. Another small negative is that they have no acquired immunity or red blood cells or vessels. So, certain diseases you most definitely cannot look at in flies. Then C. elegans is this small worm that's found in the soil, basically everywhere. They are about 2 to 3 millimeters in length. Um, they have a very short life cycle, so they're very good to work with in terms of doing genetic screens. You will retrieve mutants very quickly. The imaging possibilities are very good. Like the fish, you can mount the eggs of these guys under a normal light microscope and follow the divisions as they happen. They are very genetically amendable, which is a very good combination to have with the imaging possibilities. Basically, you can feed these guys RNA that will interfere with a gene function. You can have bacteria that express RNA that will interfere with the genes in the animal after he eats it. So it's very easy to perform knockdown studies with these animals. And they have quite high-tech genetics, I would say. They are not as high-tech as flies. Flies have always led the forefront in terms of pushing the genetic tools, but they are not far behind. A big plus is that the lineage of every cell in the animal, it's about 1100 cells in the animal, the exact lineage is known and the divisions occur in a very stereotypical pattern. They are cheap to run in the lab and they can be frozen. So that's a positive. The negative is the same as perhaps with flies, that they're an invertebrate and their relevance to certain human diseases would be questioned. There can be very valid experiments done that are related to human diseases, looking at the function of different pathways and proteins, but it still can be considered as less than optimal relevance. So these models have been helpful to figure out the mechanisms of pattern formation, morphogenesis, growth, developmental timing, and neural development. And we will touch upon some of these things throughout the time in this course. But as I mentioned, and I'll keep mentioning, they're also important in understanding human diseases, including cancer. And I will keep reiterating this, that cancer is basically a reinitiation or a re-expression of developmental programs and developmental genes at a time that is not suited. Yeah, so this isn't on the test. I just wanted to mention a little bit more about flies. So I mentioned Thomas Hunt Morgan before. He was the guy who performed the third experiment where he not only killed one of the focus cells at the two-cell stage, but he removed that cell. But he then wanted to find a better model system for his lab. And the story goes that he had this lab in New York, and one of his students left out some bananas, and the next day when he came in, his office was full with fruit flies. So he had a look at them and decided that this could be the possible model organism to work with. So there's an element of chance when it comes to picking models to work with at that time. And he received then a Nobel Prize for some of his work on fruit fly in 1933 for the discovery of genes and their placing on chromosomes. And his lab, he set up the first fly lab around 1910, I think. And he had several very successful students from his lab that also received the Nobel Prize at different times. These are shown here. You can read this if you have time. But these are not the only Nobel Prizes that fruit fly research has won. It has additionally won prizes for discoveries in the field of odor receptors and the functioning of the olfactory system, the activation of the innate immunity system, the toll system of receptors was won by Jules Hoffman in 2011. And I said that the flies, they don't have the blood system of the humans, but they have circulating macrophages, for example, and they have an innate immune system related to, say, the complement system in humans, at least related in terms of function. And then the most recent Nobel Prize was won by Hall, Robash, and Young, and this was for molecular mechanisms that control circadian rhythms. So these are the mechanisms that control day-night rhythms in animals, including us, right? Why we get tired when it gets dark and why we wake up when it gets bright. These guys did work in fruit flies that described this, and the molecular mechanisms that they found were conserved into other species. including us. And here's another slide on current animal models. Most of this I have covered already with the invertebrates, but I just wanted to mention some of the other models like the sea urchins and the ascidians and tunicates, because the guys from the SARS maybe will take them up. And these are models that are less well described, and they are involved in pushing those models forward. And then some of the vertebrate models that I haven't mentioned include chick and frog, and these have been particularly suitable for analysis of the early stages of embryo development and gastrulation, these movements, because they are these large eggs and they undergo these cleavages and movements that are found in all vertebrates. And the chick, I haven't mentioned, I might as well mention now, is a pretty good experimental model. Again, they have a very large egg. You can open the shell of the egg and you can observe the embryo as it develops. And these have been particularly suited for studies of limb and brain development. Back to the axes in the body, the anterior, posterior, dorsal, ventral, and left-right axes. We haven't mentioned too much, but just wanted to point these out in the human and say that these other animals, obviously they have these axes as well, right? The fly has a anterior, posterior, front and the back. It has a dorsal and ventral, an up and a down to its orientation. And it's divided in the middle with left and right, although they look more symmetrical perhaps than in humans. Zebrafish too has anterior, posterior, dorsal and ventral. We even call it like the dorsal fin or the ventral fin. And C. elegans, even though it's a round worm, it can be difficult to tell the head from the tail, but it does have a head anterior and a tail posteriorly. And more difficult to tell, they have a dorsal and a ventral, so they have some structures that are found ventrally that are not on the dorsal side. These animals have, in common with the human, segmented body plans: head, thorax, abdomen. Even in the not so obvious cases of the worm, it is segmented. And these repeats are, or these segments are repeated. So this is very similar to how we have a segment in the vertebrate, which looks the same in the next segment. So it's repeated over and over again. We get this extension of certain regions of the body. And this happens too in invertebrates. We can see here in the abdomen, for example, the segments are very clear. So this means these animals are suited to study the processes involved in generating, for example, these segmented repeats or the dorsal ventral polarity. I have mentioned that the animals have three body layers: the derms, and from the outside to the inside, these are the ectoderm, the mesoderm, and the endoderm. And of course, these can be found in other animals as well. And there is advantages to looking into simpler animals, perhaps, because in mammalian skin, which is the epidermis coming from the ectoderm, you have complex types of cells and arrangements, and these can be much simpler in a model organism like Drosophila. So some things, the conserved features you could study here. The more complex organizational features, a more advanced model system would be more appropriate. Of course, these animals that we talk about, they have a nervous system which also derives from the ectoderm. They have a central nervous system and they have a peripheral nervous system, just like invertebrates. They also have systems of muscles, kidney, blood in the mesoderm. It can be found in the fly or the worm. So these are suitable models to study the development of these organs and structures. They also have an endoderm. Here we see the Drosophila gut, and of course it's very small, but it has similar cell types to being found in the human gut. So it's not just the structures there, but the cell types that are important for function can also be found there. And the fly has other organs that correspond to human organs like the lung, pancreas, and the liver, and their development can be studied there as well. Again, we can show how conserved the structures and the genes are by doing experiments here. So we have a gene, PAX6, which is important for human eye development. If you lack one copy, you get these ocular defects. The eye is malformed. We could do studies in mouse to show that this was very important, this gene. And if you remove 2 copies, you get a lack of eyes in the animal. And this was true for fly as well. When we find the homologous gene, which was called eyeless in flies, because when you mutate the gene, it has no eye, but it's the PAX6 gene. Then this is just shown here with the fly again. What we could do with the fly was we take this eyeless fly and we make another transgenic, we introduce a transgene to the animal where we bring back the Drosophila eyeless gene or PAX6. And now we restore the eye. So this is the mutant for eyeless, but we bring in a copy of the eyeless gene and we can restore a normal looking eye. And we can in fact take the eyeless gene or PAX6 gene from different animals, including the mouse, and introduce it back to this fly lacking eyes and lacking the eyeless gene, and he will get his eye restored. This is called a rescue experiment. This shows how well conserved the process of, or this gene's function in the process of eye development is. Let's talk about developmental genes and control of gene expression. When we say developmental genes, we mean those genes that are important for the development of the animal. When we disrupt those genes, we end up with developmental phenotypes. So either the development of the animal stops or it gives rise to a misshapen, misformed animal. For example, we should bear in mind that we talked about there is a maternal contribution to the egg from the mother, and that these maternal proteins and RNA come from developmental genes, but they're contributed by the mother, so maternal developmental genes. Later in development, when we had a multicellular egg, zygotic transcription kicks in. So the animal itself, its own transcription kicks in of zygotic developmental genes. And these developmental genes mainly encode for gene regulatory and signaling proteins, and they're involved in the control of specific developmental processes, as have been shown by studies with mutants that we'll discuss next. Nowadays, when the genome of whole animals have been sequenced, we can compare these genomes of multicellular animals to unicellular organisms, for example, yeast, eukaryotic organism. And we can see what genes are present in both the higher organisms and the lower organisms. And those genes we can call sort of housekeeping genes because they're not multicellular. Yeast, for example, then they don't have developmental genes to give rise to this ordered multicellular animal, right? So we can also get a clue to what are developmental genes by comparing genomes. Of course, not all of those genes that are present only in the multicellular animal will be developmental genes. They can be doing other processes that the yeast doesn't do, but this can give it a clue. Then typically you go in and look at the expression of those genes using techniques like in situ hybridization to see that the genes are turned on or contributed by the mother, that the RNA is present in the developing animal. That can give you a good clue that it plays a role during the development. And then the gold standard to determine that it's a developmental gene is mutate that gene and see the phenotype that develops in the animal. In general, a particular developmental process may depend on a large number of developmental genes. So you have more than 60 genes that are involved in the patterning and segmentation of Drosophila. And it's been determined that around 9-10% of the 19,000 genes in Cenerovditas elegans, C. elegans, are involved in development. So approximately 10% of the genome in that simple worm are involved in development, so developmental genes. So a large proportion of the genome is dedicated towards making the animal, and that makes sense. Let's talk a little bit about the regulation of developmental genes, and there are several points of regulation. If you turn your attention a little bit to this figure on the right, we have transcriptional control of developmental genes, obviously switching these genes on and off. And then there are several steps before we get to the generation of a protein. You have the generation of the mRNA, its export and its translation into a protein, the modifications of the protein to give the functional protein that will do the job of the gene. But we can also mention that there are genes that are transcribed that do not code for proteins, and they give rise to, for example, ribosomal RNA, tRNA to make the proteins, in short, non-coding RNA. And these are important for development. The time it takes to make them, they may be housekeeping genes, but you need to also switch these on. So these would give developmental phenotypes, but they would be in housekeeping genes. But there's recently been the discovery of a category of regulatory RNAs, microRNAs, and in humans they've identified over 2,500 different microRNAs, and these are acting as inhibitors of translation, and they can induce targeted RNA degradation on certain genes, and these have been shown to play developmental roles in some cases, particularly in C. elegans controlling developmental timing. And while I mention timing, I just want to say that, of course, the timing of development varies a lot between different animals. And in part, that can be attributed to the amount of time it takes from when a gene is switched on to when it gives rise to the mature protein. And we can even be talking about the same protein in different animals, but maybe the protein is slightly longer or slightly shorter. That will give different time to make it. Small difference, but every step of the way here can have a small difference in the timing, the amount of time it takes to transcribe, to process, to transport. All these things can be different between different animals, and this will contribute to the length of time it takes to make that animal, or the gestation period, or and onwards, growing from the infant stage to a full adult. And not only this, of course, there are other things that contribute to the time it takes to make an adult animal. The cell cycle, we already mentioned that the cell cycle is different between different species. In some cases, the cell divisions go really quickly, and in other cases, it takes up to a day per cell cycle in the case of humans. And then the size of the animal must play a role also in how quickly or slowly development will go, with quick animals, or smaller animals developing more quickly, at least in the embryonic stages, because they need less cells, right? Makes sense. How do we identify these developmental genes, or genes that are important for the regulation of development? How do we identify them? And typically that is through mutant analysis. So we generate mutants in genes, and we can do this in two different ways. So the sort of classical way has been to apply DNA damaging agents, which will make random damages in the DNA in different genes, and we try to give a certain dose that we believe that we're only going to mutate one gene per animal that we harvest in the end. And then we look for phenotypes in those animals. So we screen for those phenotypes. And this is called mutagenesis, when we feed the chemical, and screening, when we look for the phenotypes. And this has been done in genetic models. We identify the developmental mutants, and then we try and identify the gene responsible via a range of techniques. And this is referred to as forward genetics, the examination of the genetic cause of an altered or abnormal phenotype introduced by a chemical mutagenesis or mutation by irradiation. So yes, you can use X-rays also to generate breaks in the DNA that will some of those breaks will occur in genes, and then you map the breakpoint to identify the gene. And then you perform sequencing around that, and you get the sequence of the gene. This has changed somewhat since the publication of whole genomes from 2000, the year 2000 onwards. And now we basically have the genome of all the model systems and other systems that are less common, and we can do what's called reverse genetics, where we know the sequence of the gene already and we do a targeted approach on that gene to make the mutant. For example, generating knockouts. We could use, you've all heard of CRISPR-Cas9 system to make targeted knockouts, but also we can perform RNA interference, where we generate RNA that's complementary to the region of interest and will knock down the gene function. And then, of course, we look for developmental defects if that's what we were screening for. These approaches can be used to screen for other things as well, metabolic defects or whatever. So this is the approach that has been largely used for model organisms that are widely used, but the other organisms that are less studied but are interesting to have a look at, especially for comparative developmental biology, where you want to look at the evolution of development or developmental genes. Then we can look for, so the whole sequence of these animals is not available, the whole genome sequence, but we can look for similar genes by using techniques such as cross hybridization or PCR strategies, which depend on the fact that DNA will bind to DNA, and the tightest binding will be to fully complementary, or RNA will bind to DNA, and the more complementary, the tighter the binding. But even if you have some mismatches, so of course the genes between distant animals, the more distant the animal, the less likely they will have conservation on the nucleotide level. But there will still be some, and this approach has been used in the past. So, for example, the very famous homeotic or homeobox genes were first described in Drosophila, and then they used these cross hybridization techniques to find the Hox genes in vertebrates. I mean, once you identify that a gene is present in that animal, a related gene to the one you're interested in, then you can start designing PCR primers to amplify the gene, and you will eventually recover the sequence for that gene, even though you don't have the full genome sequence. So it is possible. And from there, you can design more tools. You can make the CRISPR-Cas again on the basis of your newly recovered sequence. You can make a CRISPR-Cas constructs to target that gene and knock it out, and then assess, does it have a developmental role? This is just an image of the homeotic homeobox genes in flies that first identified. This one was called bithorax, the bithorax complex, and that's because you had this fly that gave rise to a duplication of the thoracic region of the animal, giving rise to two thoraxes and two sets of wings. And this is actually a transformation of a region of the fly. He normally has a small little haltere here, which is transformed to a full wing. And these homeotic selector genes control anterior, posterior compartment identity. And we will go through this in detail later on. I just want to show there's other types of studies you can do. When you have identified a gene, you can see what happens when I express it in the wrong place. So again, if you take a homeotic selector gene that's normally expressed in an area of the thorax where you have the legs developing and overexpress it in the head, you get the antennae that are transformed into legs. This was one of these classical experiments to show that it's involved in the identity of compartments in the animal. Okay, so we have made mutations either using forward or reverse genetics. We have our mutants, and now we need to characterize them genetically to use pre-existing definitions to describe the effect of the mutation or how the mutation acts. And the general classifications are listed here. I'll just go through them briefly. So we can have recessive mutants, which are loss of function, where the phenotype is only visible in homozygotes. So you need two mutant alleles. And an example of this is given here for the recessive mutation vestigial. So here we have a normal fly where it has two wild type copies of vestigial, VG plus wild type on both chromosomes. If we have a homozygous mutation for vestigial, VG minus, we still have a normal looking fly because it has one copy of the gene, VG plus vestigial, that is sufficient to supply the protein to give a normal looking fly, no phenotype. But if we were to cross these heterozygous flies, 1/4 of the animals would be homozygous for vestigial, getting a vestigial chromosome from the mother, a vestigial chromosome from the father. So we get a homozygous vestigial mutation. And this guy shows the phenotype with having very small wings. So that's recessive loss of function. We can have dominant mutations. It can be gain of function. So mutations that make it more active than normally, make the protein more active than normally, or constitutive active when it shouldn't be. And these phenotypes can also be visible in heterozygotes. So just having one copy of this extra active protein in the end gives rise to a phenotype. But you could also have two copies, two mutant alleles, and it either gives you a worse phenotype or it could even kill the animal, again, be lethal. Now we have semi-dominant, where one normal allele is partially sufficient and causes a weaker phenotype in the heterozygotes. And if we look over here, we have a mutation, a semi-dominant mutation in the gene brachyury, which is involved in tail length in mice. And we see again the normal mouse here with the long tail. It has wild type plus copies for the brachyury gene. In the heterozygous mutation, indicated by T here as a brachyury mutation, the animal has a shorter tail. So it's visible even though it's only lost one copy of the gene. But if we were to breed these animals and recover homozygous mutations in 1/4 of the animals, it would be embryonic lethal and we wouldn't even see the animals coming out. We would have to open up the animal to look for the missing animals. And these kind of mutations can be said to have a dosage effect. A certain dose is needed to keep the animal alive. One copy is enough to keep the animal alive, and if you lose both copies, you have no dose of the gene. The animal doesn't make it. Finally, we can have dominant negative mutations where we get a mutation in the protein coding part of the gene, which gives rise to abnormal protein. And this protein, maybe it is normally binding to itself, forming homodimers, and we get the generation of the abnormal protein and the normal protein. And when they form homodimers, those homodimers are non-functional, right? Because you have one part is non-functional of the homodimer. There's one way in which just having mutation in one allele can interfere with the other allele, right? Giving a dominant negative effect. And here we have a more detailed overview of the brachyury semi-dominant mutation And the genetic crossing scheme that I mentioned, where you get 1 in 4 animals being homozygous lethal for the brachyury gene. We can just briefly go through it. Here we have the wild type animal that is 2 copies of the wild type brachyury gene, whereas here we have a short-tailed brachyury heterozygote animal. 1 copy of brachyury is mutated, indicated by the T. When we cross these together, we will get this chromosome T, which can pair with one of the female chromosomes, wild type chromosomes, to give us a heterozygous for T and a wild type chromosome. This T can also pair with the other wild type chromosome from the female to give us 2 animals that look the same, heterozygotes, or the wild type chromosome from the brachyury heterozygote can pair with either of the wild type chromosomes from the female. See here? And we get 2 wild type brachyury genes. So these animals then have no phenotype, but the heterozygotes have the phenotype like here. If we want to get to see the effect of this recessive gene, we need to cross now the heterozygotes to recover a homozygote for brachyury. So again, the brachyury gene mutant indicated by the T can pair with either of the chromosomes from the sibling, and this will give heterozygotes. The wild type chromosome can pair with either chromosome from the sibling, giving us a potential wild type, or the mutant chromosome pairs with the other mutant chromosome, giving us the homozygous brachyury animals, which are then embryonic lethal and don't make it. Okay, just like to say a few words on the generation of mutants, induction, and then screening for phenotypes. Typically, this, as I mentioned, can be done by chemical or X-ray methods. One of the chemicals of choice has been EMS, ethyl methane sulfonate. And what you do is you feed this or expose the animals to X-rays. You look for large numbers of embryos and see if you recover any phenotypes. But one thing is there's steps in between, right? So you're feeding, you're feeding the adults. You can then look for phenotypes in the embryos, but most likely most mutations will be recessive. That means that you will need to cross the adults that emerge from the embryos again, and then self-cross typically, so that you can recover these recessive mutants. So similar to here. So you might have the generation. You have the adult where he has been fed something. His germ cells need to be mutated to pass that on to the progeny. So he would have a mutation in some gene you don't know. You cross that to wild type to recover potential mutants. And those potential mutants, you want to cross to themselves to check that out, but you need to set up a stock with this guy, and from within that stock you can then perform the cross to recover homozygotes and look for phenotypes. So it's several rounds of crossing from feeding the animal or x-raying the animal and making mutations in the germline to actually examining the phenotype. And now if we think about it, if we were to do this in mice and we say we have to do 4 rounds of crossing to see a phenotype, then we're talking each step of the way will take 50, 60 days. That's quite a long experiment. So in flies, for example, as I said, we can do it it's 10 days per life cycle. So from getting the adult to getting the next adult can be 10 days. So it's pretty quick. This speeds things up a lot. So that's one of the reasons why model systems have been used for genetic screening. Obviously, there's a lot of genes, 20,000 approximately in C. elegans and I think it's about 17,500 in flies. Okay, so we're coming towards the end here now. I just want to reiterate some points. We've covered some of the basic principles in introduction. We will go through things in more detail afterwards. We have covered some of the 5 main types of developmental processes more thoroughly than others. I would mention them again here, that we have the cleavages, the cell divisions, early embryo, pattern formation, and morphogenesis, change in shape. We haven't discussed too much about morphogenesis, but it will come. I would like to mention a few words on cell differentiation and growth, which I feel we haven't mentioned at all yet. These are the 5 main types of developmental processes which overlap in time. They can be happening at the same time, and they influence one another. So just a few words on cell differentiation. It is the generation of structurally and functionally different cells. It typically occurs over time and in several steps. We have an example here of a cell from the blastomere in the early embryo up to forming a beta islet cell in the pancreas. And you see that it goes through many steps. It becomes specified to become an endoderm cell. It becomes specified to become a pancreatic bud cell, and more specified to become an endocrine pancreas cell. And from this pool of cells, we get beta islet cells. And along the way, we have many rounds of cell division. This happens over time. And we can think of this differentiation. The cells are becoming more and more determined. They're losing their potency. The blastomere cell in the embryo is totipotent. It can become anything. The early endodermal cells are like stem cells and are pluripotent to become any type of endodermal cell. But at the end, the beta islet cell, it's terminally differentiated and it has no more options. It can only be that. And this is a type of cell determination, and it's a one-way system towards the terminal differentiation. And during development, the potential possible fates of the cell become progressively more limited or restricted, as I mentioned. And the cell, during its life, it's not sitting in isolation. It meets a set of cues from its surrounding cells and undergoes irreversible changes that limit its choice for differentiation, right? In another way, the cell has a memory. It remembers what it was before and the cues that it has met along the way. And then we didn't mention growth much at all, so I thought we should say some words on that. And it occurs mainly after the fundamental body plan has been generated and the organs have been specified and created. And then we get this round of growth and expansion, shown here how things are growing and changing. It involves cell divisions, cell growth, and the deposition of extracellular material, for example, bone and connective tissues. These are all involved in the growth of the animal. It can contribute to morphogenesis because differences in growth rate can change the overall shape, and we see the baby as it's growing up to be an infant, or the embryo as it's growing up to be an infant. And we see that if you look at the proportions of the body at this stage, the head is obviously much bigger than the final form. So the rest of the body grows. The head grows more first, but then the rest of the body grows afterwards. So this is growth that's contributing to change in shape. So, okay, we're sort of right at the end here. And then normally in a live lecture, I would remind you of the learning goals that we set out to cover today. And I could do it here as well, but with focus on what could potentially be asked in the exam. So we have identified the central questions in developmental biology that has driven the evolution of the field, with new questions or new answers to questions leading to new questions. Yeah. You should, for the exam, become familiar with the core processes of development and the terms that we use to describe them. And this part was just an introduction to that, and we will continue with that with different model organisms. You should appreciate the historical conceptual advances made in developmental biology, but that's not so important for the exam. Okay, but some of the practical approaches that were used to advance those theories, like grafting and killing off cells, and what those experiments led to in terminology when it comes to developmental biology, this idea of mosaicism, for example, that's important to know. And then I spent a lot of time describing different model systems, the common ones that are used, uh, and the pros and cons of those. And those would be important to know for the exam. And then I gave you a cell cycle refresher. Um, for the exam, at least we won't go through the cell cycle. We're not doing that in depth here, but at least know what's the difference between meiosis and mitosis, right? How do we generate germ cells and what's going on there? We spent a bit of time on that, and that could be good to know for the exam. And then we spent some time talking about genetics, crossing, and at least you should know how to define different types of mutations, right? Whether it's recessive or dominant or semi-dominant, gain of function, loss of function. You should know that there might be questions on that in the exam. And I point out that these are all potential exam questions. We will be generating 2 exams. Um, so we have to generate many questions, and half the questions will be in one exam, the first exam that you take, that I hope everybody takes, and I hope that everybody passes. And then we have half the questions left over for the repeat exam, which I hope we don't have to use. Okay, that's the end of this lecture. Talk to you again soon. Bye.